Welcome, everyone, to the North Atlantic Fire Science Exchange's Fall 2025 Student Webinar Series. My name is Eric Evanson. I'm the Science Communications Specialist with NAVSE. Now, the goal of the Student Webinar Series is to connect students with the fire and forestry community to develop positive working relationships as well as exchanging knowledge. And speaking of exchanging knowledge, just want to remind everybody that the North Atlantic Fire Science Exchange, or NAFSE for short, is part of the Joint Fire Science Program's Fire Science Exchange Network. You can see on the map there are 15 exchanges all across the country. And if you want to learn more about any of the exchanges or the Joint Fire Science Program itself, we encourage you to go to firescience.gov. So for today, our speaker is Shyrill Mariano from the State University of New York at Albany, or SUNY Albany for short. And her presentation is going to be Transformation of Manganese During Vegetation Burning. Now, the way the session will go is Shyrill will share her presentation first, and then we'll follow up with a question and answer session at the end. Now, if any time during her presentation you have any questions, feel free to pop those into the chat, and then we'll visit those at the end of the session. And then we'll follow up with a little update related to our upcoming talks that we have in the remainder of our Fall 2025 Student Webinar Series and ways that you can get involved. And so with that, I'm going to stop sharing and I'm going to hand it over to Shyrill, and she can start her presentation, and look forward to you sharing with the group a little bit about yourself, your background, any other connections that you might have with the fire and forestry community. So take it away, Shyrill. Thank you.
Yeah, thank you, Eric, for the introduction. So good afternoon, everyone. Thank you for your interest in my talk, which is the transformation of manganese during vegetation burning. So a short background on me is that I have a degree in geology and marine science, so I've been really attuned to the earth sciences since before. This picture, let me show the laser first. So these pictures are from my previous experiences in fieldworks. And from where I come from, wildfire science really isn't established. So when I got into the PhD program here in U Albany in the Environmental and Sustainable Engineering Department, it was a new field for me. It's just that the analysis that I have been doing before are also applicable to the research that I'm doing right now. And I have been learning a lot, and it's really an interesting topic for me, especially that fire itself isn't used as an ecosystem management tool back home only for agricultural practices, but here I learned that it's used for ecosystem management. And I hope to contribute to that pool of knowledge with our fundamental research. So my focus is on nutrient cycling impacted by natural and anthropogenic disturbances, so in this case, wildfire. And one of the questions I usually encounter when I present this research is that why did I choose manganese, or why are we focusing on manganese? Is it really that important? So in order to fully appreciate the role of manganese in the ecosystem, we look at the bigger picture, which is nutrient cycling. Here we see a simple diagram of the nutrient cycling process, and it's the transfer of energy and matter between the living and non-living parts of an ecosystem. We get nutrient inputs from the atmosphere, from the soil, and it also involves both above ground and below ground components. And manganese especially plays an important role in the decomposition process. Manganese is not as abundant as, let's say, calcium or nitrogen, but manganese is involved in various parts of this nutrient cycling. And in decomposition, it helps to release nutrients stored in the litter, which these nutrients are then used up by existing and regrowing vegetation. And now we'll look at manganese cycling closer. In the environment, manganese exists in different oxidation states. It exists as Mn2+, 3+, and 4+. And how manganese participates in cycling processes strongly depends on what form it is existing. As manganese 2, it is the most stable and most soluble form of manganese, and this is the form of manganese that is easily taken up by plants. It is released by soils through the weathering process. In the right environment, it gets oxidized to manganese 3+, which is relatively unstable, very reactive, so it does not last long in this form unless it becomes incorporated in mineral structures or is combined with organic ligands. Further oxidation of manganese 3 plus forms manganese 4 plus, which is the least soluble form of manganese, but it is very reactive. And this reactivity drives the redox cycle back to Mn3 plus or Mn2 plus. And all these manganese forms are present in the diverse group of minerals we call manganese oxides, which are composed of manganese, mainly composed of manganese and oxygen in the structure. So this fourth picture here is a basic structure of a manganese 4 plus oxide, but this is just one of the many structures of manganese oxides present in the environment. If we look at the manganese cycling within soil plant systems, we see that manganese exists in different parts as different forms. Manganese 2 is taken up by the plants, is stored in the leaves mainly as manganese 2 still, and also manganese 2 in the fresh litter. Since it's easily soluble, then it is released from the litter, and the microbes in the soil and the litter itself oxidizes manganese 2 to manganese 3, and further oxidation forms manganese 4 plus. And this manganese 4 plus and manganese 3 plus later form solid manganese oxide minerals, which whenever they are in the soil, it interacts with the soil organic matter, which drives back the cycle to manganese 2 plus, which then again, it can be taken up by plants or it can be leached out from the soil if it's not immediately taken up. So this is the process of manganese cycling without fire. Now, what happens when there is burning? We know that fire affects other ecosystem processes. It is a natural occurrence. And we know that this is not necessarily a bad thing. That's why we expend extra effort understanding how fires mesh with the other ecosystem processes we know so that we can be informed and create stronger science-based burning practices. The most noticeable effect of fire, of course, is that it induces soil carbon loss through the combustion of organic matter. High severity fires can also render enzymes inactive and deplete the microbial community. And this effect specifically impacts the decomposition process, which I highlighted before. Ecosystems, especially the mature ones, when they run out of manganese from the weathering process, they rely on the recycling of nutrients from the release of these nutrients in the biomass, which occurs during the decomposition process. So if fires affect the decomposition process by affecting the microbial community, then the pathway of decomposition is affected, impacting nutrient cycling, the soil nutrient status, and overall the carbon cycle. Nutrients can also be lost by fire through volatilization and particulate transport in smoke emissions. And for nutrients that are not easily volatilized, like let's say calcium or phosphorus, they are physico-chemically transformed. And this transformation affects how they return to the soil and how can they be used again by regrowing vegetation. For example, a previous research in our group showed that calcium and phosphorus at high temperatures are altered to insoluble minerals. So then these nutrients are unavailable for immediate plant uptake. But whether this is a good or bad thing still requires a lot of investigation. So for my topic, we focus on manganese, and we know that manganese plays an important role in soil plant cycling. So this leads us to ask, how does fire transform manganese, and how does this transformation affect the processes that manganese is associated with? To answer questions, we have analyzed wildland fire ash samples from various ecosystems, and also we did additional sampling from the Albany Pine Bush Preserve, particularly for prescribed fires in a wood-dominated and grass-dominated part of the pine bush. And also we collected biomass samples for laboratory simulated burning. To determine if burning duration, burning temperature, and initial chemistry affect manganese transformation, we had this setup for a simulated burning. We burned white pine needle and black spruce, and also a pure organic manganese compound to test the effect of initial chemistry in manganese transformation. We controlled for time by keeping the temperature constant at 550, and we burned from 5 minutes, 20 minutes, and 5 hours. We also controlled for temperature, burning at 450 and 600 degrees Celsius for over 5 hours. And the ash generated from this simulated burning is analyzed through a technique we call X-ray absorption spectroscopy. I won't go into the nitty-gritty of this technique, but the principle is that we subject the ash sample to X-ray, which excites the element's core electrons, and the X-ray energy required to achieve this is very unique. So it's very sensitive to the elemental species, and we obtain a the spectrum such as this, which we can be divided into two regions: the XANES, X-ray absorption near spectroscopy, and the EXAFS region, extended X-ray absorption fine structure. So these two terms, I will be repeating it a lot through the rest of the presentation, since most of our results focus on these two spectra. Generally, the higher the oxidation state of an element, the more energy is needed to excite the atom's electron. And thus, the spectra that we get for the XANES, this edge and the peak moves to the right, the more oxidized the element is. This specific figure that I'm showing here are actually from manganese references. The blue here is manganese 0, and the purple spectra here is from a manganese 4+ reference. So later you will see similar lines to this one, and the more this rising edge and the peak is to the right, the more oxidized our sample is. And we estimated the average oxidation state, or the AOS, of our sample by fitting our sample with references published in literature, and we computed the contribution of each reference to that spectra. EXAFS, on the other hand, tells us the relative distance of another atom from our central atom. So imagine the manganese is here at 0, and these shells, these peaks, tell us the relative position of the nearest atom. It could be another manganese, it could be oxygen, it could be another element. And so the whole EXAFS spectra gives us the idea of the structure, the chemical coordination of our atoms in the mineral. So what did we see in our ashes? For the wildland fire ash samples, generally we see that all our environmental ashes are oxidized compared to, let's say, fresh litter, which would usually have an oxidation state of 2 to 2.3, but our environmental samples from 2.5 to 3.3. And you can see here in the spectra, we have shaded regions. The green one is where we expect the manganese 2+ peaks to be, and the orange and red shows us where we expect the manganese 3+ and manganese 4+ peaks should be. And the more oxidized our ash are, the more these peaks are in the right side. So we know that manganese in the ash is oxidized. So what determines the oxidation state at which the ash manganese forms? What is clear is that long-term burning for high temperature at 550 and long-term at 6 hours, we observe complete oxidation of manganese from 2.3 to 2.7. They completely oxidize to between 3.8 to 4. Now what happens if we change up this temperature and burning duration? First, for the burning duration, we see from the XANES spectra that the longer the burn, the more this spectra is to the right, the more oxidized our ash is. So longer burns for the white pine needle ash resulted to a complete oxidation to 4+, whereas those that were burned between 5 and 20 minutes were just yielded 2.7 and 2.8 average oxidation state. EXAFS spectra confirm the differences in the speciation between the 5 hours and the 5 and 20 minutes. Manganese oxides in general are characterized by an oxygen in the first shell, which we see in all our samples. But differences start at the second and the farther shells. For the long-term burning at 5 hours, we see that the second shell is associated with manganese, and the third shell shows calcium. Meanwhile, for the 5 hour and 20 minutes, we still have oxygen at the second and manganese at the third and fourth. And so this confirms that the species between these ashes are different. And actually, for the fitting of the XF spectra, for the long-term burning, we have a calcium manganese oxide reference, which is a manganese 4 plus species. Meanwhile, for the 5 minutes and 20 minutes ash, we used a manganese 3 plus reference. So this XF spectra aligns with our Zane's result. In terms of initial chemistry and burning temperature, when we burned the pure organic manganese compound at 450 and 600, we yielded two different manganese minerals, which is Mn508 for the 450 burning and Mn203 for the 600-degree burning. In contrast, for the white pine needle ash, we obtained almost the same spectra with just a slight difference in the estimated oxidation state. And when we try to identify the species, we get almost a similar structure for our mineral in the white pine needle ash. And if you think this is familiar, this is the same calcium manganese oxide that we have fitted from the previous experiment with control on the burning duration. So why is this calcium manganese oxide a point of interest for us? Let's go back a bit to the undisturbed manganese cycling, which is driven by microbial decomposition. The microbial decomposition of the microbial oxidation of manganese yields this mineral MnO2 or birnasite. And birnasite is a manganese 4 plus species. It's the most reactive 4 plus species, and it is abundant in decomposing lither. On the right, you can see its structure. The red ones denote the manganese, and the purple ones denote the oxygen. If we compare that with the manganese oxide that we're seeing in our ash, we see a slight difference in structure, in the geometry, and also, of course, the glaring difference in the inclusion of calcium in the calcium manganese oxide mineral. And though the differences seem subtle, these small differences can already mean a lot in the reactivity of the mineral. So birnasite is known to be very reactive, but there are less studies with this calcium manganese oxide, specifically when it's present in the environment. And this is what we're targeting to focus on for the next part of our research. But we already conducted an initial reactivity experiment just to test just how reactive our ash is and how does the transformed manganese potentially affect that reactivity. So we used two ashes, one from the prescribed fire and one from the simulated burning. And note that the prescribed fire is less oxidized compared to the simulated burning. And we reacted that with an organic compound that is commonly found in soils just to simulate an environmental condition. And after reaction for 18 hours, we tracked the degradation of this compound, the cathedral, using UV-vis spectrophotometry. So we plotted the ratio of the absorbance after and before the reaction. And we can see here that the APBP, the prescribed fire graph, has higher ratio, meaning it's less degraded compared to the white pine ash, which is from the simulated burning. And so we can see that the mass loading as well affects the degradation of the organic compound. And from this graph, we also demonstrated that aside from the reactivity influenced by the increasing mass, the oxidation itself shows an effect on the reactivity of the ash. The more oxidized the ash is, the more it degrades our organic compound. So in summary, we now have some answers to the questions I posited earlier, and it's that manganese is differentially oxidized depending on the burning condition, whether it's the temperature or the duration, and also the initial chemistry of the material. So it's interesting to note that between two different biomass, the black spruce and the white pine, we're getting the same mineral, the same manganese minerals during the same burning conditions. And also, we demonstrated that ash with oxidized Mn species are capable in degrading cathedral, which is an organic compound usually found in soils. And this suggests their redox reactivity and their potential role in post-fire soil cycling. And the implications of this in our post-fire environments is that the immediate transformation of manganese compared to, let's say, the microbial-driven oxidation suggests a shift in the manganese redox reaction rates and pathways. Consider the amount of biomass that undergo combustion, particularly in high-severity fires, and the enrichment of manganese in the ash compared to later. So fire introduced a significant oxidized manganese pool in the soil, and this might change the paradigm of organic decomposition on the forest floor and topsoil, particularly when the burned environment is recovering. Moreover, the control of burning temperature and duration may have practicable applications in prescribed burning. We may have to consider what type of manganese we're getting if this is affecting post-fire decomposition processes, because then we can investigate if this change affects significant short-term and long-term influence in our prescribed burning objectives. So overall, we hope that by furthering our understanding of how fire transforms ash nutrients and how it affects the ash's role in post-fire biogeochemical processes, we can also improve our understanding on the response of ecosystems to fire disturbances. And that is all. I would like to thank my mentor, Dr. Wisham Wang, and our collaborators, and the national laboratories from which we conducted our analysis, and also our funding providers, NSF and New Albany, and also we thank the Albany Pine Bush Reserve for being generous, allowing us to sample for our studies.
All right, well, thank you, Shyrill. If anyone has any questions, this would be a great opportunity to put them in the chat. And I've got just kind of one follow-up question for you, Shyrill. Is so those samples that were in the laboratory, when you did the laboratory simulation burning, that those samples were from that map where you showed all across the globe. Did were there any samples done like after a prescribed burn, or is these are just everything is before there was either a wildfire or a prescribed burn?
We did have samples after a prescribed burn, specifically for when we conducted the reactivity experiment. So this APBP sample is after a prescribed fire in Albany Pine Bush Preserve in a wood-dominated area.
Okay, great. All right, thank you. Yeah, that answered my question. We've got a question here. Let's see. Oh, all right, two questions. So from Shlomia, first question, besides decomposition, what do plants or other parts of the system do with manganese, and how does the oxidation state affect their ability to do it? We'll start with that one.
Okay, so aside from the composition in the plant itself, manganese is actually a cofactor of calcium during photosynthesis. But it's still in manganese 2 plus oxidation state. But the interesting part for this is that remember when we did a simulated burning experiment, our manganese oxides are calcium manganese oxide. So this highlights the relationship between calcium and manganese in the foliar region. Yeah. And in the biomass, generally manganese is 2 plus. So yeah, if it's in another state, it's not then incorporated in the plant structure itself.
All right, I hope that answers the first question. And the follow-up is, how long does the higher oxidation state of manganese due to burning last? What causes it to convert to a lower 2 plus state, and over how long does that happen?
Is this during the burning, or wait, sorry, let me just.
If you want to look in the
okay. So we haven't tested how long does the high oxidation state from burning last, so I cannot answer this accurately. But yeah, but we can test that maybe later because it might have implications for reactivity.
Okay. Thank you on that one. Is there any more questions if people want to put into the chat? All right. So with that, Shyrill, if you want to stop sharing your screen, I'm going to share mine for just a couple minor follow-up things here. This. All right. Just kind of wanted to let everybody know that Shyrill kicked off our fall 2025 student webinar series. We really appreciate that. And we still have 2 more sessions in this series. You can see on the screen there. We've got a talk from Sam next Thursday, and then the last Thursday of the month, Maya will be presenting. So if you want to get more details related to those two talks, you can either hit the QR code on the screen there, which will take you to our NAFC student webinar series page, or you can also go to our website, same place, firesciencenorthatlantic.org, to get more information and registration information related to those 2 upcoming talks. And then I will say this, if you or someone you know has a project that they'd like to share in this type of format, please reach out to me. Again, my name is Eric Evanson, and my email is nafchelp@gmail.com, and you can send me an email or have the person get in touch with me, and we'd love to have them. We've already got a couple speakers for our spring 2026 student webinar series, so the more the merrier. That'd be great. And then I do want to mention that when the browser closes, when you end this session, there'll be a very brief survey that pops up, and it's 4 really quick questions. So if you could do that, fill that out, we'd greatly appreciate that. And it looks like Shalomia has her hand raised. Did you want to put something in the chat real quick?
We have a question in the chat, actually.
Oh, okay. If you want to go ahead. Is it for me or for you, Shyrill?
For me. So
Okay.
is there a minimum time that would be recommended to obtain positive results for MN? At this point, we don't have that information yet, but that is what we are hoping to investigate in the next part of our experiments.
Okay, great. Yeah, I was going to say that that's kind of all that I had. And so I just wanted to thank you, Shyrill, for your informative talk today. And it was greatly appreciated. And I appreciate everyone joining today to listen in. And we're going to go ahead and keep the session open so that if there are any more last minute questions, we'll leave this open for another couple of minutes, and we'll welcome any more feedback and discussion on Shyrill's topic. And with that, just thank everybody again for showing up. Remember, we've got a couple more in our fall series. We hope to see you next week and the week after. And otherwise, thank you again and have a great day.